Hi, I'm Donna Chang, CEO of Hope Biosciences, here to talk to you today about optimization and standardization in stem cell manufacturing for the fight against COVID-19. So first off, a big thank you to Thermo Fisher and Gibco for this very special invitation. Just by looking at all your backgrounds and your talks, I just feel surrounded by greatness and innovation and invention and the amazing advances in stem cell research. The topic of my presentation is based on stem cell manufacturing. And although it's probably not as exciting as the pioneering work that you all are doing, I hope I can provide some insight to the latter stages of clinical development. Now, traditionally in drug development, we begin with an idea, and then we sort of work from the beginning in the lab and in vitro assays, and we do some kind of proof of concept work, then preclinical models, and then into clinical trials. But after approaching therapeutic development in this way for over a decade, about 4 years ago, I decided to start Hope Biosciences to try it a different way. And I took it completely backwards. So I'm married to an IT person. So in the IT world, when you build software, you have to start with the requirements. Because if you don't, you will find yourself constantly reworking, correcting, reprogramming, troubleshooting forever. So if we take this approach into drug development, we have to start with the requirements. So let's start there. So in clinical trials, your requirements are really the study design. Now, before we begin the study design for a COVID-19 trial, all I have to say is that COVID-19 is unlike anything we've ever seen, and that is an understatement. This disease, the real difficulty with this one is the sheer speed at which the disease manifests itself and the speed at which, you can be okay 1 minute and then all of a sudden, basically on your deathbed. And the speed, it makes it incredibly challenging to treat. So it makes, for us, all other diseases that we treat as challenging as they are, like things like ALS and TBI, Alzheimer's, and all of our other trials, it actually makes those look a lot easier because those are chronic degenerative diseases. So by definition, you kind of have time to deal with those symptoms and those conditions. But in COVID, if you're going to attempt to use a cellular therapeutic to fight COVID, you're going to need to deliver this often, so fast, and you're going to need doses that are very large to be able to combat a disease like this. So it's a very difficult task. If you look at the design for our hospitalized, this is for the hospitalized cases of COVID-19. For those of you who are interested in reading more about this trial, if you go to clinicaltrials.gov, it's NCT number 043262189. This particular study is a very quick one. If you look from enrollment to end of study, you're looking at only a 28-day period. And within that 28-day period, you have to deliver 4 treatments of 100 million stem cells within a 10-day period. So every 3 days, you have to be able to deliver 100 million cells. Now, I'm not going to be discussing the mechanism of action of MSCs. That's an entirely different talk. But just in a nutshell, the idea is that we deliver these cells intravenously. They go into the body. They help with immunomodulation. So as we know in COVID, we have an overreacting immune system. So we try and modulate the immune system and also help with lung regeneration. Okay, so I'm going to add to this a little bit and add some complexity to this, meaning we're going to add some more requirements. If we're going to deliver these cells, we want to deliver them fresh, okay? Because if we're going to give MSCs to combat this virus, then we have to know they're at least viable and healthy enough to be able to combat it. So they have to be fresh. Though this trial was strictly for allogeneic purposes, it could theoretically be autologous if a person had their own cells banked, and it could be allogeneic, so using a donor source. Now let's say we use a donor source. Well, FDA says if you're going to use a donor source, you're going to have to use the same donor for all 4 infusions, okay? Because we don't want to introduce any other, HLA compatibility issues here. So we have to give the same donor 4 times. All right? Is that complicated enough? Okay. So on top of that, let's talk about what the challenges are in really executing this. There are technical challenges, but there are also business challenges. And from my seat here, I kind of look at the overall picture of where we are. And when I look at the industry as a whole, cell therapies for any kind of diseases are expensive, particularly autologous therapies. That sort of tends to, we sort of price ourselves out of everything because it's just too costly and it's not scalable. You also have stability issues. You know that stem cells have terrible stability, so that doesn't really work in our favor. Donor-to-donor variability. Now that's a real kicker because everybody has different cells. And if you were to attempt to do autologous treatment, imagine how everyone's cells behaves differently and everybody has different rates of growth and different personalities and all of that. So the consistency of your drug product, because these are drug products, at the end could be variable, and that just cannot happen, right? So in addition, you also have potential cell integrity issues because we know that long-term cell culture can cause all kinds of problems too, not just in cell characterization, but function, all of that. And then all of you people who are in the lab, you feel me when I talk about the woes of culture, stem cell culture, like over time, your cells will get enormous and they'll start sticking to each other. There's an overproduction of ECM. You're going to get dead cells floating around. I mean, it's just those problems are also out there. So how has the industry tried to solve all of this? Okay, how have we approached it up until now? Well, I can tell you that we have largely, and I talk about us as an industry as a whole, we have seen many companies go towards, going to bioreactors, doing large production runs in bioreactors, making thousands of doses. Benefit of that is you use a donor or a group of donors to create this one lot. Good thing about that, you only have to QC that one lot one time. So that's great. And then you put it all in a cryotank. So you're essentially storing it for long-term storage. And that creates that kind of stability, if you will, because it's at least stored in liquid nitrogen. Well, when cells have to be used, you would just take a vial out of that tank and thaw it and give it to the patient. But we actually, before we tried anything, we tested this approach because we wanted to understand what this actually looks like. Because traditionally, thawing cells from a cryotank didn't have a great viability, but we knew that technologies have gotten a lot better now, that post-thaw viabilities are actually pretty good. So we put it to the test. This is data where we took, you'll see 8 different pictures here, 4 on the top and 4 on the bottom. And this is using a freezing solution. Basically, this is what it looks like in the hemocytometer. We're just counting cells here to see what the viability was post-thaw from cells that were stored in HB101, which is our standard MSC growth media that we produce in-house, along with 10% DMSO. And we stored these for a week. Why a week? Because that's what we've done so far. I mean, we are looking at long-term, more longer-term studies, but for now we're looking at just a week. So they weren't frozen for that long. They were thawed, and the viability was checked immediately after thawing. And we found that the viability was around 76 to 78%, which is very good. We were very surprised because it was much better than what we've normally seen. To confirm whether these cells were truly viable, we decided to reseed them. And we did this primarily because we couldn't find any existing data out there of anybody who had tried to do this. And a lot of times we judge the health of the stem cells, and these are all mesenchymal stem cells, adipose-derived. We judge the health of these cells by how well they adhere and they're able to grow, okay? So when we reseeded them, we found that the actual attachment rate was between 18 and 20%. And that is when they're grown in HB101. They're inoculated again in HB101, which is the media that we know our cells can thrive in. So that begged the question, what happened to those 80% of those cells? Are they just not healthy enough to adhere and grow? Well, so we thought maybe the freezing solution, there's something wrong with that. So we tried 10% DMSO, 1% human serum albumin, and sterile saline. Viability, we checked. It looks like 70 to 71%, so about the same. And actual attachment rate, 18 to 24%, a little bit better, but still not so great. So we looked at maybe human serum albumin might have something to do with it. So we changed it to 0.1%, and we found that the viability upon thaw was a little less. And what was really surprising is that we found actually no attachment at all after we took those cells and reseeded them. So that told us that we have to find a different solution. And so when you look at the control group, which is just fresh cells, never been frozen, when you harvest the cells, you'll see after harvest 94 to 99% viability. If you were to take those cells and reseed them, you'd see the attachment rates from 89 to 90%. So it's close to the actual viability. So we know that, for us at least, this made sense, that there is a need to deliver fresh cells. But how? How are we actually going to get this done? That's where our solution comes in. So we have developed a way to be able to deliver fresh, undifferentiated, autologous or allogeneic. We use allogeneic cells only in the case of there's no autologous donor. Culture expanded mesenchymal stem cells on demand. And the on demand part is what's important because we can not only expand these cells, but we can do it whenever it's needed. So we can repeat the same process for the same donor over and over and over again when it's needed. So if you go back to the requirements, the study design, when you see how patients are receiving more than one infusion and they're supposed to be receiving the same product repeatedly, this is where this becomes very important. So let's start with cell culture. What are the issues with cell culture and how do we get over these things? So one typical thing that changes during culture is morphology, and people don't really think this is a big deal because stem cells tend to keep their shape and their form. But one very important thing to note is that stem cells during culture will start to grow physically larger, and it's just a tendency that they have. Also, a note for all of you is that our product is always released at passage 4. So it's a passage, we go from P0, from fat directly to P0 to P4 at release. Why am I showing you data up to passage 12? You're going to see data throughout this presentation going up to passage 12. FDA asked the same question. Why do you give us data up to passage 12? Because it's very important to show consistency in the process. That though we continue, though we always stop at P4, the point is that it can be continued. And there's value in knowing that because you know that your product will always be consistent. Now, look at cell size. In all of the morphology here, even through passage 12, you notice that this doesn't change. So here we go in real time. Well, this is fast forward, time-lapsed here. But cells on the left are being grown in HB101 in our proprietary media. And then on the right, you have DMEM and 10% FBS. Our media already contains 5% FBS. So you can already see here, as they begin to proliferate, you see on the right-hand side that the cells are slightly slower and they're now physically getting larger. They didn't start off that way, but they start doing this pretty quickly. Now, for those of you who are technical in the lab, actually doing hands-on cell culture, know exactly what we're talking about here, that you can't grow completely to confluency, because if you do reach high confluency rates, you're going to end up having extremely sticky cells that you would need a spatula to try and get off of your flask. So that is one of the classic problems with cell culture. So maintaining cell size, the actual integrity of the original cell, is very important. So in a short period of time, if you look at our cell growth period, typically, if we're growing in this rate, we'll probably seed 1 million cells, and in about 5 days, we can reach about 1.5 million cells. With our process, in 5 days, we can achieve about 32 million cells from 1 million cells. Now, you're probably saying, okay, she's presenting the best outcome, but I have proof. We had a clinical trial. Our very first clinical trial was on rheumatoid arthritis patients. We had 15 patients come in. It was an open label study, so they all received their own cells. Average age, 51. Average disease duration, 15 years. These people are very sick for a long time. And you see that the cell morphology on all of these patients is pretty much the same. The color's a little different based on the microscope and the lighting that day, but otherwise everything's the same. This is so important because when you're working on clinical trials and you have a clinical outcome that you want to prove, like what happened, how did this happen, you have to always go back to the product. Because when there are inconsistencies in the product, there will likely be inconsistencies in the clinical result. That's why this is very important. It goes back to, what did these people receive? Why did they see this clinical benefit? Oh, well, we can see they all received a similar benefit because they all received a similar product. So not only from a morphological standpoint, you also have cell characterization. You have to know, what their cell markers are. And for those who are part of, development in drug products, you know that in autologous therapies, at least, you have to release every single lot is its own release. So you'd have to run FACS on every single exit that you do. And we maintain cell markers and across all the passages too. If we have to go past passage 4, we don't, but for any reason, if we needed to, that we can maintain that throughout culture. Differentiation, we do this in validation to show that the cells actually still have their differentiation capacity, because after all, we have to know that the cells are going to behave the way they're supposed to when they get into the body. And we know that they can become all these different lineage even at later passages. So we know by this evidence that we're adequately culturing these cells to maintain their stem cell function. Maintaining genetics is very important. We test this through karyotyping as well as CGH analysis, which we're still in the process of doing. And we are looking at even the minute changes, like, are they significant? What do they mean? And we're looking to make sure that the genetic integrity of the cells are maintained throughout all these passages. Now, this is something that this is a CPDL, which is cumulative population doubling. When you look at the population doubling in cell culture, it's normally all over the place. And it's all over the place because it's never exactly consistent. And that's sort of the difficulty, yet the beauty of natural products, right? Natural stem cells are going to have their own personalities and do what they want. So, but if you were to think about, okay, what is the future of cell medicine going to really look like? Then, you have to be able to tell a patient that on next Tuesday at 4 o'clock, you're going to get your stem cells. Remember the requirements. If you go back to the original study design for COVID, it's every 3 days. So if you're going to release, QC clear it, and then get it on an airplane, let's say you have to FedEx it overnight, there is no room for error. Your exit day, your harvest day, has to be exactly the right day and time. Otherwise, you're not going to make it. And when you think about cell production, that seems almost like an impossible feat. But if you somehow are able to create a linear or a predictable growth, then you know you can get this right every time. So something that we've been working on over the years. So the last thing that I say is actually the most important out of all the factors is viability, because viability is what I believe is a direct indication of the potency of the product. After all, if your cell is dead, then it's not going to do anything, right? Simply said. So maintaining viability is key. Our cells, upon harvest, will remain viable for 4 days at 2 to 8 degrees Celsius, which is cold temperature, which is refrigerated temperatures. So this means that nationwide shipping of fresh cells is actually a reality. And in some of the clinical trials that we're doing, we're already doing that. We're FedExing overnight to different places, and cells are being infused. So now that we have understood the cell culture process, let's put all of this together. Okay, the cell expansion part is done, but now how are we going to deliver cells to meet the requirements that were posted at the beginning? Okay, so it all starts with a tablespoon of fat, and you probably wonder why a tablespoon of fat out of all things. Well, I can tell you that in America, we have really no shortage of fat, and everyone seems to be really happy to give it to me. So that's why fat is used. We could get MSCs from anywhere, but this is really the easiest place to get it from. And we can get it from children and adults, sick, not sick. It really doesn't matter. Pretty much everyone can part with a tablespoon without any major issues. So once this fat arrives at our lab, we will isolate the MSCs. We'll start with about 100,000 MSCs from that fat tissue. We discard everything else. It's just the MSCs that we're looking for. So it's a small amount, but that 100,000 gets seeded. We wait until it becomes totally confluent at P0. We'll bank some of those cells. We'll go on to P1, bank some of those, and passage again to P2. P2 is the last passage before all cells are banked. This is what we call a master cell bank. So each individual gets a master cell bank. Okay, that's cryopreserved, and we go through the freezing process. Why? Just to buy us time, so that we have all the time in the world to use these cells however way we want to. Okay, so now let's go to making the cells. So if we go back to our clinical trial design, 10 days before the first infusion, we're going to take out a vial. This is a P2 vial. We're going to take it out. We're going to seed a flask. We're going to go to P3 and P4. So we culture fresh. Then we harvest. And we can do this. We can repeat this up to 1,800 times from a single fat extraction, from a single tablespoon of food. We can of fat. We can repeat this 1,800 times at a 200 million to 400 million cell dosage. Okay, that's approximately 360 to 400 billion cells from a single tablespoon of fat. So that is how we are able to repeat this, and that is how donors that have their cells that are banked can use them to not only treat COVID, but we have prevention trials as well. We have 2 prevention trials for COVID. Those are also being used for that. And if you think about the future and what these cells could be used for in the future, the same cells that are banked could be used subsequently for future conditions and even for prevention. And that's what makes this whole idea of cell therapy so incredibly exciting. So when we send our cells to the hospital, we do it with a lot of faith that the research and the time that we've put into our technology will prove to be helpful. However, we are also very much aware that the very opposite may happen, and we may find that MSCs are not as effective as we thought. But whatever the case might be, we hope that the data that we generate will provide clear answers by eliminating product-related variabilities. So hopefully some of the things I've shared with you today will save you some time and heartache as you go through development. So best of luck in all your research. Thank you.